That's good. And then you get a Christmas vacation. Holiday vacation, yeah. So that'll be nice. Hopefully, maybe get to catch up on a little bit of sleep at that point. All right, so this morning we wanted to go ahead and kind of continue where we left off last week. So last week we had left off talking a little bit about both what we call the contractile or the mechanical events in the heart. We looked at this kind of alternating system of diastole and systole, right? And we saw that that's how the heart pumps blood. Then we looked at how we can actually detect the electrical events of the heart. We looked at the ECG. And we saw how those two things are very much interconnected, right? Our intuition, I think at this point, tells us that when we have the depolarization of a heart chamber, the thing that's going to follow is a contraction, right? So those two things are, the cardiac cycle and the ECG are very closely connected with each other, right? So they aren't, even though we look at them separately, they aren't two separate things. They're very much interconnected with each other. And that's kind of where we left things last week. I want to talk just a little bit today in lab about those worksheets that I gave you guys from the BioPack to work on, and then we'll kind of go from there. But we do need to start talking a little bit about blood flow through our blood vessels today, right? So that's kind of where we're at now. We've now talked about where the blood is pumping blood out of the heart, and it's got to go somewhere, and it's going into our blood vessels. So we want to talk a little bit about our blood vessels today and the blood flow through those. Before we do that, I have a little bit of stuff I want to kind of finish up with the heart and the performance of the heart, and then we'll kind of dive into the performance or the blood flow through the blood vessels. All right? Are there any questions before we get started? I feel like we're missing some people. It feels like a small group this morning. All right, let's go ahead and get started then. So if there's no questions, then I'll assume that we're all good for what we talked about last week. So I wanted to go ahead and just kind of dive in. So the topic that we touched on very briefly last time that I said that I wanted to expand on today is this concept of cardiac output. Okay. Cardiac output. We touched very briefly on this last time because I needed to make a point. But I wanted to expand just a little bit more on it today. Remember, we abbreviated cardiac output as CO, and at least for now, that's going to be our abbreviation that we use for that. Cardiac output is equal to. You remember what we said it's equal to? We only mentioned this in passing. It's equal to our heart rate times something called our stroke volume. We may not have even mentioned this. Cardiac output is equal to your heart rate times a variable called your stroke volume. Okay. We know what heart rate is, right? We know that in a given period of time, it's the number of times that your ventricles contract, right? Usually we measure it in terms of the number of contractions per minute, okay? But stroke volume is, and probably a new one to us, it's going to be the volume of blood that the ventricle pumps out with each contraction, right? So this is the number of beats per minute, or contractions per minute, beats per minute. And this is usually measured as the number of milliliters of blood pumped in a single beat, okay? So in other words, how much blood is the heart pumping with each beat? Now, if we think about it, we kind of need to consider both of those variables to really say anything about how effective the heart is, right? Because the heart could be beating really slow, and you might say, that person's really, healthy. Their heart isn't beating very fast during exercise, right? But if it's not pumping very much blood, then it's not really that effective of a pump, right? So we need to understand both of these variables in order to really talk about the effectiveness So let's go ahead and run through with some sample numbers and see what we come up with for sort of a basic cardiac output. So if we plug in some numbers here, I'll use green for that. If we plug in some numbers here, say our average resting heart rate is 72 beats per minute. Uh-oh, notes hit the floor. And our average stroke volume is 70 milliliters per beat. Okay, so 72 beats per minute, about 70 milliliters per beat. If we do the math on that, if I was right years ago when I wrote these notes, it gives us about 5,040. What are our units here? Okay, gives us about 5,040. We see that beats cancel each other out, and that gives us milliliters of blood per minute. Okay, so about 5,040 milliliters per minute, or we can round that a little bit to about 5 liters per minute. Okay. Okay. Do you know how much blood you have? About 5 to 6 liters, yeah. It obviously depends a lot on body size, right? People with a smaller frame are going to have less blood than people with a larger frame. And of course it varies quite a bit on height. We're going to see that tall people obviously have more blood than people who are a little bit shorter. But let's just say that on average you have about 5 liters of blood. Okay? What this tells me is that essentially at rest, because both of these are largely resting numbers, at rest you're pumping about your entire blood volume in 1 minute. That's a lot of blood. Okay? Think about that. It's five of your water bottles per minute of blood that's getting pumped through your heart. Okay? That's pretty significant. During exercise, this number can go up. Okay? How could we manipulate these variables to have cardiac output go up? What if we increase stroke volume? Heart rate. Is cardiac output going to go up? Yeah, right? Because that number gets bigger. What if we increase stroke volume? Is cardiac output going to go up as a result of that? It is, right? Yeah. So during exercise, both of those numbers increase. The sympathetic nervous system causes both of those numbers to go up. We know how it causes heart rate to go up, but we'll talk about stroke volume in just a couple of minutes. But during exercise, this number can go up to 30 liters a minute, or 35 liters of blood per minute. That means it's pumping your entire blood volume somewhere between 6 to 7 times per minute. Let that sink in just for a second. Your entire blood volume, 6 to 7 times per minute. I don't know what a liter of blood weighs, but a liter of water weighs 2 pounds. Okay, so if we go with that, 2 pounds times 30, that means that your heart is pumping 60 pounds of blood per minute is what it should be pumping during exercise. That's significant, right? That's a lot of stress on one organ to be able to pump that in a really efficient way. It's really a remarkable organ in a lot of different ways. But anyway, at rest, we're looking at a cardiac output of about 5 liters per minute. We can assume that that's our entire blood volume, approximately once per minute. Okay. We know that you have, these sympathetic, parasympathetic effects on your pacemaker cells that increase or decrease your heart rate. We talked about that a little bit last week. What about stroke volume? How are we causing stroke volume to change? Okay. How is stroke volume changing? Okay. This one's a little bit more nebulous, a little bit more conceptual. I think heart rate is a nice one because we see that we can affect ion permeability with either norepinephrine or acetylcholine and either cause an increase or a decrease in the heart rate. But stroke volume is a little bit of a goofier one, and it's a little bit harder for us to sort of visualize. So one of the things that we do know is we know that when stroke volume increases, so does the force of the contraction. Okay? And when stroke volume decreases, so does the force of the contraction. So a generalization that we can make is that there's a proportionality between contraction force and stroke volume. They're directly proportional to one another. So when stroke volume goes up, so does contraction force. Okay? When stroke volume goes down, so does contraction force. Okay? So one goes up, the other one goes up. They're directly proportional to each other. So if we think about the filling of the ventricle, okay, and we talked about this a little bit last week as well. Now your ventricle's like this sac, right? This sac that you're filling up with blood. And in that sac, you've got actin and myosin fibers, right? So in that sac, you've got your sarcomere. Let's go ahead and kind of draw one up on the board right now. You've got your sarcomere right here. You've got your thick filaments kind of right here. And then you've got your thin filaments that are kind of overlapping with it, right? Right, you've got your thin filaments that are sort of overlapping with it, like this. Right. Right, so we've got a situation kind of like that. Of course, this repeats itself. So we have our light bands and our dark bands that we learned about, right? So what happens is as we fill up the heart with blood, what it does is it causes this to sort of separate just a little bit, right? It pulls it apart, right? We're stretching out the muscle fiber when we fill the ventricle with blood, okay? I don't know if whoever you had for 50A talked about this or not, but let's talk about sarcomere length, right? How long is this thing? Let's talk about sarcomere length and strength of skeletal muscle contraction, because this is a much easier one for us to sort of think about, right? So let's just say, for the sake of argument, that I'm at the gym pumping iron. I know that's hard to believe. I don't actually do that. Don't get any ideas. But let's just say, for the sake of argument, that I am, right? And I've got my dumbbell. I'll use this gallon of water that's been sitting here since last academic year. I don't even want to know what's growing here. But let's just pretend that this is my dumbbell, right? Turn it on the side. Let's just say this is my dumbbell, and I'm doing the heavy curls, right? I'm doing the big ones today. I've got the big guy out. For those of you who know this, and you've lifted something in your life, so you can probably relate, right? Even whether it's a weight or not. Is it easier to get that good curl on if you start way down here with that sarcomere super stretched out? That's A, that's option A. Is it easier to get a good curl if you start way up here and just do that? Or is it easier to start somewhere like right in here? Yeah, right? That's like the easier place, right? So there's like an optimal sarcomere length for getting the maximum force of contraction. If you start way down here, it's almost like you have to get over a ledge before you can get it up. You know what I mean? Proverbially. So anyway, so there's this optimal sarcomere length. Cardiac muscle is no different. There's an optimal length where it gets the most force of contraction. If we underfill our ventricles, we're not going to get that much force of contraction. And similarly, if we overfill our ventricles, we're not going to get as good of a force of contraction. So just like with Goldilocks and the Three Bears, there's sort of a maximum or an optimal stroke volume where we get the maximal force of contraction. Okay. Okay. So contraction force is partially determined. Contraction force is partially determined by by sarcomere length. Again, too much, or it's not going to be that great. Too little, it's not going to be too great. There needs to be there's a sweet spot there. Okay. All right. So the other one that also determines contraction force is a little bit of a goofier one, and it's called the contractility of the heart. This is where things get a little nebulous, so bear with me for a minute. The contractility of the heart. The contractility of the heart is defined, let me come over here. The contractility of the heart is defined as the ability of the heart to contract at a given sarcomere. Okay. The ability of the heart to contract at a given sarcomere length. Okay, so what the heck does that mean? Okay, let's go ahead and kind of break this down a little bit. And I think the best way to do this is sort of graphically. So I'm going to erase my sarcomere, and we'll draw a little graph right over here to kind of break this down and explain it to you. So the first thing that I want to look at is I want to look at force versus stretch. Let's put an axis on the board. Okay. And we're going to look at force of contraction versus the stretch of the ventricles. Remember, stretch is basically sarcomere length, so we'll just go ahead and put that here in parentheses. So what we normally see is, as we just described, right? As I start to stretch that sarcomere out, right? I'm up here, I'm gonna use my biceps as an example, even though we're talking about the heart. I'm up here and my sarcomeres aren't stretched at all, okay? The force that I'm gonna get is pretty minimal, right? Because it's all the way up here, right? So when my stretch isn't very much, my force isn't very much. But as I start to put blood in my ventricle, we see that we have sort of an increasing force of contraction. An increasing force of contraction, and that goes and goes and goes. Now eventually I can continue to put blood in the ventricle, and the force is not going to increase after a certain point. It kind of levels out. And this is essentially what we have when we compare force and stretch. It's largely intuitive. It largely follows what we just said, where as we put blood in the ventricle, the heart is going to increase more and more and more forcefully. But eventually, like I said, we do sort of hit this point where you can keep adding blood to the ventricle, and you're just not going to get any more force out of your contraction. Okay? The good news is that, generally speaking, anything above this range is outside of what we would actually encounter in the human body. It's outside of what we call our physiological range. Okay? So within normal physiological, like everyday sort of a scenario, what we're looking at is everything to the left of this, and we see an almost nice, perfectly straight line. Again, to recap, the more blood you put in the ventricle, the more forcefully it's going to contract. Okay? So, as stroke volume increases, the heart contracts more and more and more forcefully. Okay? You guys have definitely felt this. After exercise, how do you feel? Your heart, you can feel your heart beating harder, right? It's contracting more forcefully when you exercise. Okay? This curve that compares the force of contraction to ventricular stretch or ventricular filling, this curve is called a Starling curve. called a Starling curve. Okay. Who do you think discovered it? I'd be really surprised if it was Smith, right? But there was a physiologist named Starling who discovered this. In fact, what we're talking about right now, this entire concept, is called the Frank-Starling law of the heart. And ironically, his name was not Frank Starling. I don't remember what his first name was. There was another physiologist named Frank who also helped him to discover this relationship. But the curve is called a Starling curve. Okay. So, as stroke volume increases, the heart contracts more forcefully. What about this idea of contractility of the heart? Okay. What about this idea of contractility of the heart? Again, this is defined as the heart's ability to contract at a given fiber length. So, get this out of my way here. Let's go ahead and draw a second curve here on my same axis. Let's say that we have something like norepinephrine or epinephrine now binding to receptors on the heart. What we might see is we might see a Starling curve that looks like this. Okay, so this is one where we see increased contractility. How do we know that? Let's go ahead and pick a fiber length. I'm going to go ahead and just choose a fiber length like this one right here. Okay, so I've stretched the ventricle out. I've put some amount of blood in the ventricle. If we follow that up, you see that we intersect our first curve right here, and we come all the way up and we intersect our second curve right there. If we draw a line over to the y-axis for both of those, we see that at this fiber length, my initial Starling curve had this much force of contraction. But my new Starling curve with increased contractility had this much force of contraction. So we saw that at one fiber length, the heart contracted more forcefully. There was increased force of contraction. Okay. So that's an example of increased contractility of the heart. So that's increased contractility that we see there. Okay. Does that make sense? Sort of, yeah. Give me one sec. I will use that as an example, yes, but just let me, I'll finish that thought too. But the norepinephrine, epinephrine do increase the contractility of the heart. Okay. Okay. So, the question was norepinephrine, epinephrine. Do they do this? They do. They do. Epinephrine and norepinephrine do indeed have an effect on increased contractility. Okay. So epinephrine and norepinephrine increase the contractility of the heart. Okay. In fact, any chemical that increases the contractility of the heart can do this. And there's a number of chemicals. We call them inotropic agents. Okay. So a chemical that affects the contractility of the heart is called an inotropic agent, and specifically one that increases the contractility of the heart is called a positive inotropic agent. Positive inotropic agent. So there's a whole bunch of different drugs and things like that that can cause the contractility of the heart to increase. We find a lot of plants have chemicals that are inotropic agents. We don't really have it in Southern California, but the one that comes to my mind almost immediately, there's a flower up in Northern California and the Pacific Northwest that tends to grow fairly prolifically. It's called foxgloves. Does anybody know what foxgloves are? You see them occasionally like at Home Depot here, but they a stalk, and they look like little mittens. They're like little purple and white or red and white mittens that you might imagine a fox might slide its paw into, which is how they get their name, foxgloves. They're really, really pretty. But like I said, they don't grow terribly well in our climate. We're much too hot and dry for them to really be good. But they have a chemical in them called digitalis. Okay? They have a chemical in them that occurs naturally called digitalis. And digitalis, like epinephrine and norepinephrine, are a positive inotropic agent. One of the things that they say, and this, I don't even mean to laugh because it's mildly morbid, but they tell you if you have small children at home that you shouldn't plant foxgloves in your garden because little kids like to eat things in gardens and stuff like that. I mean, I'm just going to speak for myself. When I was a little kid, I used to eat things out of my mom's garden all the time. I'd pick leaves and flowers and green tomatoes and things like that and eat them out in the garden. And usually I would get into trouble because the tomatoes were still green and didn't need to be picked yet. But kids are just naturally curious and they put things in their mouths, right? The thing is, there's so much digitalis in foxgloves that if they eat enough of it, it can increase the contractility of their heart to such a point that it actually gives them a heart attack. And that's not a good thing for them. So we try to avoid that in gardens where we might run into curious children and things like that, right? Because we don't really want that to occur. Digitalis is often given as a drug as well for patients with weakened hearts because it does increase the contractility, causes it to beat harder, and it can increase their stroke volume as well as their cardiac output as a result of that. So it's an important one. Okay, so stroke volume, to kind of recap, is a little bit of a goofy variable. It's hard to look at directly. It's hard to measure directly. To truly measure somebody's stroke volume, you would need to actually put something in their ventricle that can measure the pressure changes when it fills and when it empties. And to do that, you usually have to thread that in through their femoral vein. So you have to go in through here and you have to go all the way around to their ventricle. And it's more than just measuring heart rate, which you guys could measure right now very, very easily just by finding a pulse somewhere, right? So it's a lot more involved. It's a lot harder to measure. So true cardiac output is actually kind of difficult for us to measure because this is a tricky variable for us to get at. And it's pretty invasive. But we can make some generalizations about it based on what we do know. And we know that it does, when stroke volume increases, we know that contraction force increases, and we can look at some of the chemicals that are going to affect that and see how that sort of works. So when you exercise, for instance, what's happening is you have epinephrine affecting not only your heart rate, but you also have epinephrine and norepinephrine causing an increase in contractility of your heart, and that's going to, by default, increase your stroke volume. So we see that we have those sorts of things occurring. So it's a little bit more of a nebulous variable, but it's definitely one worth considering when we talk about the performance of the heart in general. Okay. All right. So cardiac output. Any questions about that? I know specifically that your lab manual asks some questions about Starling curves, so I wanted to make sure that we went over those this morning. Okay. Well, if
You said that stroke volume was the nebulous.
It's not as obvious to us, right? Like heart rate, like I said, you can measure it right now. It's really like you almost don't even have to think about heart rate to get an idea of what it is, right? Stroke volume, harder to measure, a little bit harder for us to sort of understand the factors that affect it because it's just not as intuitive to us. It's kind of all I was saying, yeah. All right, let's go ahead and turn our attention then to the blood vessels, okay? So once our ventricles contract, what they do is they pump blood either into the pulmonary trunk or the aorta, and subsequently to a number of other blood vessels through the body. Let me pull up a picture really quick. Take a look at it here. Starling curves are fun exam questions. What's happening? There we go. All right, there we go. All right. So our heart pumps blood into our arteries and ultimately through a series of blood vessels, right? So let's go ahead and just take a quick look at this. We'll talk a little bit more about the anatomy of arteries and veins today in lab. So I'm not going to go into that too much right now, but just to kind of give you an idea of where our blood is going, let's go ahead and kind of take a look at this. So again, we're pumping blood out of our heart into our arteries. So this is an example of an artery right here. And again, we'll kind of do a compare and contrast of arteries and veins in lab today. So I'm not going to go too far into it right now. But those large arteries, like the aorta, are going to branch into smaller and smaller arteries. And some of those arteries are still pretty big. You know, they're carrying blood to our limbs and things like that. So they're still fairly significant arteries. But they do branch and they get smaller and smaller and smaller. Eventually, kind of our smallest arteries are these ones called arterioles, and they're a lot smaller than our large arteries. Those arterioles eventually branch into capillary beds, which are our smallest blood vessels of all. Your capillaries far outweigh or outnumber your large arteries. You've got tens of thousands of these capillaries running through your body. And they're significant enough that just about every cell in your body is within a cell or two of a capillary bed. So they permeate almost all of your tissues, and they're incredibly small. They're small enough that your blood cells have to pass through a single file. So they're very, very small little passageways. But of course, this is where we have the exchange of oxygen and nutrients and things like that. And then on the other side of it, we see that your venules ultimately are catching the blood from those capillaries. Those are all going to converge into veins, and those smaller veins converge into larger veins, like your inferior and your superior vena cava, and we ultimately are returning that blood to your heart. Okay. So again, we'll expand on this whole idea a little bit more today in lab. We'll take a specific look at the anatomy of arteries and veins today in lab. But suffice it to say for right now, with some exceptions that we'll also talk about in lab, probably on Friday, our circulatory system follows this pattern where we go from arteries, arterioles, to capillaries, to venules, and ultimately back to veins, and then back to the heart. Okay, and it is significant, all right? All said, you have roughly 50,000 miles of blood vessels in your body. Okay, if you were to take them all out and lay them end to end, you have about 50,000 miles of blood vessels. Most of them are capillaries. Okay, most of them are capillaries. All right, your aorta, your largest artery, is only about maybe 2 or 3 feet long, and that's a long way from 50,000 miles. So we have to find that distance somewhere, and most of it comes from our capillaries. So significant number of them in your body, and they have to be dense. They have to be compact, because we have to be able to fit them into a vessel of defined size. That's you, and just about every cell in our body needs a capillary very, very close to it, and we'll expand on that idea as well in lab. So it's a significant number of vessels that we're talking about. Okay, but this is the delivery system for oxygen and nutrients that we see, and it's really important that we sort of understand some of the concepts that are involved here. I think the first concept that I'd like to talk about is just the dynamics of blood flowing through blood vessels. And the nice thing about this is that to understand blood flowing through blood vessels, we can sort of gather a lot of knowledge from just physics, okay? Because the blood flowing through your blood vessels is subject to the same physical laws that water flowing through a pipe is subject to, okay? Or water flowing through a hose or something like that. So if we look at the plumbing in your house, we're going to see that the same physical laws govern all of that, as does blood flowing through blood vessels, all right? So that's kind of a helpful thing. And we're going to see that that's true with the respiratory system as well, which makes me really happy because it's less that we sort of have to remember. So let me put the screen up. Let's talk a little bit about the dynamics of blood flow. So if I have a blood vessel, or a pipe in your house, use a section of it right here. But if I have a blood vessel and I have blood flowing through that blood vessel, okay, like this, I see that I do have a forward moving component to that blood, right? There's something propelling that blood forward. In our bodies, that's the heart that's creating the pressure that's going to propel that blood forward. If we look at the plumbing in your house, there's a water main somewhere, and that water is being pushed from some distant upstream pump that's pushing that water through your house, right? So we have this forward moving component to our blood. But the blood is not just doing that, it's constrained to this tube. And as a result of that, we see that the blood is also pushing outward on the wall of the tube, or the liquid is pushing outward on the wall of the tube. It's not just blood, it's any fluid in any tube is pushing outward on the wall of that tube. Okay. We call this pressure, okay, in fluid dynamics, we call this pressure the hydrostatic pressure. Okay. Hydrostatic pressure is what we call this in fluid. Hydro, of course, is referring to water or fluid, I guess. And then static refers to the fact that it doesn't really move, right? As long as the tube remains intact, that fluid doesn't really go anywhere. It's just pushing outward on the wall of that tube. Okay? Now, problems arise when the tube fails, right? When this pressure exceeds the strength of the tube, that's when problems arise, okay? This is why when you go to Home Depot or Lowe's and you buy PVC pipe for your sprinkler system or something like that, you notice that there's a pressure rating on the side of that PVC pipe. It tells you the pressure that that tube can withstand, and you shouldn't exceed that pressure or you could have problems, right? The good news is Riverside has no water pressure at all, so you're fine. But if you've ever put in a sprinkler system, you know that there's no water pressure in the city. But at any rate, you're definitely under the threshold. With blood vessels, okay, we also have this. You and I have a slightly different name for the hydrostatic pressure in blood vessels. We call it our blood pressure. Call it our blood pressure. So your blood vessels have to be able to withstand that static pressure that's being put on their walls by the blood. If your blood pressure becomes too high, or if the wall of the blood vessel becomes weakened, we stand a risk of that rupturing. It's called an aneurysm when that occurs, and that's not good. It's never good. So we want to avoid that situation at all costs. Okay. Okay. We all good so far? Hydrostatic pressure, blood pressure. Okay, so it's an outward pushing pressure on the wall of our blood vessels. Okay. It's time for another graph. We need a really long x-axis for this one. Maybe not that long. I'm gonna have to erase some of it. We're gonna look at pressure now, measured in millimeters of mercury. And we're gonna look at different parts of our circulatory system. So our y-axis is pressure. Our x-axis is gonna be different parts of our circulatory system. I'm not as concerned right now about the pulmonary circuit, so we'll focus on the systemic circuit. So I'd like to start in the left ventricle. Okay, so here we are in the left ventricle, and then we can go to our aorta and our arteries, our arterioles, our capillaries, our venules, and our veins. And then ultimately back to our right atrium. Okay, I did need my whole x-axis. Look at that. Okay, so we're basically writing down our systemic circuit on our x-axis, right? Starting with our left ventricle, our aorta, our arteries, our arterioles, our capillaries, our venules, our veins, and our right atrium. Okay. Okay. So let's take a look at the pressure that we experience or that we can measure in our left ventricle. Okay. Now, before I do that, let me remind you guys about your cardiac cycle. I'm going to try to do this without putting the screen down, just to try to simplify things a little bit. But if we take a look at our left ventricle, we remember that we're going through a cycle of diastole, or relaxation, and systole, contraction, right? And during diastole, there is a period where the chamber of the heart, essentially, for all intents and purposes, is empty, okay? It's not for long. Blood never stops flowing, so it pumps this blood out, and then it relaxes, and then it has to refill, okay? We don't need to put numbers to it right now, but what can you tell me about what you think is happening in the ventricle during, let's say, early diastole? Okay, but if you were looking at this picture, what do you think is happening to the pressure here in the left ventricle? Do you think it's relatively high, or do you think it's relatively low? Low, right? Because there's no blood in it, right? There's no blood in it to put any pressure on it. So you're right, the pressure is relatively low. In fact, even though blood never stops flowing, right? It's not like there's a measurable period of time where your ventricle is really empty. But there is a measurable period of time when your ventricle is nice and relaxed, right? The pressure is real low in there during that time, okay? But then the ventricle is going to enter systole. It's going to contract, okay? So what the ventricle is doing is it's pushing on that blood, okay? It's pushing on that blood to drive it out of the ventricle and into the aorta. So the ventricle is applying pressure to the blood, okay? And another law of physics, or another rule of physics that we learned, is that if we apply pressure in this direction on the blood is going to apply an equal and opposite pressure in the opposite direction. So when the ventricle squeezes the blood to push it out into the aorta, do you think the pressure is still low, or do you think it goes high? It goes up, right? Okay, so it goes up. So in the ventricle, we see diastole, and the pressure drops drastically. And then we see it squeezes the blood to push it out of the aorta, and the pressure goes up considerably. Okay, so let's go ahead. I'm going going to mute my screen really quick. Let's go ahead and draw that onto our graph. Okay. So in the ventricle, we see a really low pressure, and then we see a rise. Woo! Right? When we have systole that occurs, right? And then diastole, when the pressure drops. Woo! And then we see a rise. Woo! And then a drop. Woo! Right? Like that. So we're seeing big swings in blood pressure in our ventricles, from exceptionally low to relatively high. Okay? Any questions about that so far? Yeah.
So the pressure usually is up and down? Or is it
Yeah.
not always consistent?
Not in the ventricle, right? So in the ventricle, we see this complete emptying of the ventricle. And when that happens, there is nothing to apply pressure to the wall of the heart, so the pressure drops very, very low. Okay? And then it fills up with blood, we have systole, and then the pressure rises, right? So in the ventricle, we see big swings in the pressure.
Up and
Okay.
down.
Up and down, okay? But then we pump the blood into our aorta. In the aorta, the pressure changes, the story changes a little bit, right? Because does the aorta empty between contractions? Let's take a look at our diagram again here. Let's take a look at our diagram and let's think about this a little bit. No, it isn't. Of course, the thing I always want to project is right up here at the top of this, and I don't really understand that. But let's take a look right here, right? So we just had, we pushed blood into the pulmonary trunk, we pushed blood into the aorta, and now we're back in systole. Look at this, right? Your ventricles effectively are empty, right? We said that pressure drops very, very low. But look at your aorta. Is it empty? No, right? It isn't, right? Because what happens is you push some of that blood into your aorta, out into your systemic circulation. But then gravity, it drops back onto that semilunar valve, right? And so there's still blood in the aorta. So is the pressure lower than it would be during systole? Definitely, okay? But it's not zero. Okay, so in our blood vessels, the blood pressure never drops back to zero. All right, so what we see in our aorta and in our large arteries is we see a slightly different situation occurring. Okay, what we see is we see that the blood pressure comes up during systole and then it drops a little bit, right, when the heart relaxes. Okay, and then it comes up and then it drops. And then it comes up, and then it drops, and then it comes up, and then it drops. So in our aorta, in our large arteries, we see a fluctuation in blood pressure that looks something like this. We see a peak in our blood pressure during systole, okay, because we're pushing a bolus of blood out into our blood vessels. We see a peak during systole. And then we see a valley, I guess, during diastole. Okay. And when you have your blood pressure measured, you almost always are given your blood pressure in two numbers. What are those numbers? Systolic and diastolic, right? So this is essentially what we're looking at here is we're looking at the maximum blood pressure you might experience during ventricular systole, and we're looking at the minimum that you might experience during ventricular diastole. If we come back over to our y-axis over here, you'll notice that the way that I drew this is roughly to where our patient's blood pressure would be 120 millimeters of mercury during systole, and about 80 during diastole. So this would be a blood pressure of what you would call 120/80, okay? Now, if that number gets too high, of course, we can run into problems, and so on and so forth, but we have that. Okay. All right. All right. Actually, do that. Okay. Any questions so far? How many of you guys know how to measure blood pressure? All right, cool. You guys get to be the teachers in lab today. We're gonna learn how to measure blood pressure. Everybody's gonna learn how to measure blood pressure in lab today because it's a useful skill to have. Okay. As our blood travels through those major arteries, we see this rise and drop in blood pressure. Every time there's a rise, every time we have a ventricular systole, that is a bolus of blood that's sent out into the arteries. And you can actually feel that wave of pressure going through your arteries. All you have to do is find a superficial artery, like your brachial artery or your radial artery or your carotid artery, and feel. And you can feel that pressure wave going through. That's what we call your pulse. Okay? Every time we have ventricular systole, you can feel that pulse wave traveling through your major superficial arteries. Okay? And there's a lot of different pulse points. Like I said, you can do the carotid, you can do your brachial, your radial. There's a popliteal pulse on the back of your knee. There's a few others as well, but these are kind of the big ones. So take just a minute, find a pulse point, whether it's on your brachial artery, your radial, or your carotid. Just take a second and feel that pulse wave travel through. Every time your ventricles contract, you feel that wave of pressure traveling through your arteries, right? Every single time. This is why we can use pulse as a proxy for heart rate, right? Because every time there's a ventricular contraction, there's a pulse wave, okay? Now, the farther we get from the heart, the more that pulse wave slows down. So they're actually, if you were to do a little experiment where you measured pulse at different places on the body, you'd find that you'd have some slight variation because it does slow down as it gets a little bit farther from the heart. But generally speaking, this is the way that pulse works, and this is why it makes a great proxy, because it is a wave of pressure traveling through your arteries, and every time you have a ventricular contraction or a heartbeat, you can feel that pulse wave going through your arteries. All right? But what's happening here is we are moving from our aorta to our major arteries into our arterioles and ultimately our capillaries. And these blood vessels are getting smaller and smaller and smaller in terms of their diameter. So one of the things that we have to do is we have to think about what happens to fluid flow in really small tubes. Okay, because that's what we're getting to, is we're getting to tubes that are just exceptionally small. All right. To do that, we need to do a little bit of math, and I don't have time for that right now. So we'll pick up in lab with that. We'll talk a little bit today about blood pressure and stuff like that, and then we'll kind of go